So today we're going to talk about microbial genetics. So that's really how the DNA or the genome of any microorganism influences its properties. And when we think about pathogenic microorganisms, they have fundamental differences in their genetic makeup from non-pathogenic organisms. So we've all heard about new and emerging diseases like, in my lifetime, HIV is a new and emerging viral infection. Also, more recently, the swine flu or H1N1 influenza. Also, new forms of bacteria like MRSA, methicillin-resistant Staphylococcus aureus. So these are all new genetic forms of microorganisms. In the case of H1N1, it's a new form of a well-known virus, the influenza virus. Also, in the case of MRSA, it's a new form of a well-known bacteria, Staphylococcus aureus. But it's really these properties that bacteria have that I'm going to review with you today that sometimes transfer to other microorganisms, like something like a virus like HIV, which is a very fast-changing virus. Its genome changes very quickly, even within the individual that's infected with the virus. And this is all genetic changes that are going on. So today I'm going to focus on bacteria and their genetic properties and how they carry out fundamental processes such as interpreting these genes and changing them into proteins, so the transcription and translation steps. And then I'm going to talk a little bit about how genetic information can change and how bacteria can adapt to changes in their environment or selective pressures in their environment. So I'd like you to understand from Chapter 8 the key processes that sustain life. So DNA replication and also protein expression, which transcription and translation are the two steps that are required for a gene to be made into a protein. I hope that a lot of this is review for you. And I want you to be sure to know kind of the overview, the products and the key enzymes or factors, macromolecules like ribosomes that are required for these processes. There are some key differences, though, between these processes occurring in bacteria as opposed to eukaryotic cells, which you may have studied already. And I'll go over those key differences. And it has to do with the lack of compartments within bacteria, which makes them able to carry out processes like transcription and translation simultaneously. So as soon as that gene is transcribed into mRNA, even before the mRNA molecule is complete, the translation of that messenger RNA into an amino acid sequence of a protein can begin to happen because there's no separation. There's no requirement for transporting that messenger RNA out of the nucleus into the cytoplasm or onto the rough ER, where you would find the process of translation occurring in a eukaryotic cell. There are also important ways three major ways that bacteria are able to adapt to changes in their environment, whether it's the presence of a new and alternative carbon source, or whether it's the presence of a drug like penicillin. Bacteria are able to adapt to these changes in their environment through three major ways. They can mutate, so they can have small changes in their genome that result in changes in their proteins. They can share information between bacteria in their environment, so gene transfer without having to replicate or to go through the process of binary fission. Bacteria that are existing very close together in their environment can sometimes share information if they have the proper capacity. So if they have these extra information like plasmids, they can be transferred, or they can pick up information in other ways from viruses or from the destruction of cells in their environment. And then the third way, mutation, is regulation of protein expression, so turning genes on and off. So I'd like you to understand those three major ways that bacteria adapt, and I'll go over some examples. You should know how enzymes are regulated. We're going to go over an example of the lac operon, lac standing for lactose. So lactose in some bacteria, such as E. coli, if glucose is absent or has been expired from the immediate environment and lactose is present, then E. coli are able to continue to grow because they're able to use this alternative sugar in their energy production. And the way that genes are organized, some genes are organized in bacteria, is in a cluster. So rather than having one promoter for each gene like we do in eukaryotic cells, often several genes that are necessary for a certain function, like lactose metabolism, three key genes, are clustered together. They're only made as one transcript, one piece of mRNA, which then is made into the three different proteins through translation. But in this way, they can be very efficient. They can turn those genes off when there's no lactose present in their environment, or when they have plenty of glucose to metabolize, or they can turn it on when the need arises. And then there are changes that bacteria are able to use, changes in the expression of their genes or of their appearance. They can change disguises, basically, based on pressures in their environment. And in this way, a lot of pathogens are able to escape the immune response or to delay the immune response, which is based on recognition of surface molecules like antigens on the flagella or on the capsule or on the cell wall. And by changing these antigens in a process called antigenic variation or phase variation, they're able to change their disguise or form a disguise so that they can persist in the host a little bit longer before they're eliminated by the immune system. You should also know the differences between transfer of genetic information from one generation to the next, which is vertical genetic transfer, versus those that are in a population that are not necessarily dividing, so already pre-existing cells that can exchange information. And that's called horizontal gene transfer. And there are many ways that vertical gene transfer can occur, but it's basically one type of change to the genome, and that's either it's mutation. So either a single point, a single base is changed, or there's a fragment, frame shift that's lost or gained in that DNA structure. And sometimes it won't affect the protein that's produced at all, and other times it will affect the protein such that it's no longer functional. So vertical change occurs through mutations, which are either spontaneous or can be induced by mutagens in the environment. So who can think of a mutagen that we're exposed to every time we go outside?
Ultraviolet radiation.
Ultraviolet radiation, exactly. So from the sunlight, our DNA can change. And that's why going out in the sun too much can affect the DNA of our cells so much that we can get cancer. So basically it's a genetic change that the UV light is exposing, is inducing in our own skin cells that would result in that change in that tissue, resulting in cancer. So the same thing, bacteria exposed to UV light can also be susceptible to mutation through that mutagen. Now there's horizontal gene transfer can occur in basically 4 different ways. It can occur via accidental introduction of new DNA by a viral infection. It can occur by little bits of DNA that are picked up from dying cells by those cells that are capable of transferring these fragments of DNA into their cell. It can also occur by direct contact between 2 bacteria in the process of conjugation, and this requires that special appendage called the sex pilus. It makes a tunnel, a connection between the cytoplasm of each cell through which a plasmid DNA can be transferred. So this extra chromosomal piece of DNA can be transferred there. Or even part of the genome can be transferred. And then transposition is actually the rearrangement of genes because of this element called in the category of a mobile genetic element, so an element that easily moves from place to place, either within a genome or between organisms, which includes plasmids. But the element for transposition is called a transposon, and because it has mirror image sequences in it, as well as an enzyme encoded within it that will cleave the DNA, it very frequently will find homology in other parts of the genome and will jump from place to place, either interrupting a gene so that that protein is no longer made, or introducing new genes, like genes for antibiotic resistance, into an organism, such as a bacteria. So these are some of the terms that I've taken from your chapter that I expect you to be familiar with. I first want to just review what we know about the bacterial genome. So we know it's double-stranded DNA, that it usually comes in a single circular form. In the case of E. coli, it's kind of in the middle of like an average size for bacteria with 4.6 million base pairs. But the size can vary. It can be very small genome in the case of very simple bacterium, like mycoplasmin. We talked about they're the smallest of the bacteria, and or it can be very large. Something like Mycobacterium will have a genome on the larger end, like closer to 9 megabase pairs. So this genome is organized, and here's a rough map of the genome of E. coli. It will have several genes that are necessary for different functions, biosynthesis of macromolecules like amino acids, nucleic acids, lipids, carbohydrates, synthesizing peptidoglycan, components of the cell wall, lipopolysaccharide, and things like that. And it will have an origin of replication that's necessary for the replication of the entire genome. And just like with eukaryotic DNA, it has a direction. So there's a 5' carbon at one end and the 3' carbon at the other end, and it reads in the direction of 5' end to the 3' end, and that's the direction of the replication. It can replicate in two directions at once, so bidirectional replication, which I'll go over in a minute. And it's often, as I mentioned, organized into clusters of genes that are important for a single function, like lactose metabolism, so operons. Gene expression for essential genes may be constitutive. They're always being expressed, always being transcribed and translated. So those that are constitutive may be regulated by something that stops their expression, so by repression, whereas others need to be induced to be produced into their functional counterpart of the protein. So here's a fundamental diagram of the flow of genetic information that would occur in bacteria. So we already talked about binary fission as the means of replication. So there's always transfer of the entire genome during binary fission. You need to have replication of the entire genome during that process. So it's a copy of the genome. It's almost a perfect copy, a very, very low rate of change there by spontaneous mutation, basically because of the mechanisms that the DNA polymerase, the enzyme that's required for that process of replication, has these proofreading functions. So it kind of checks its work as it goes along. But changes can occur, and those occurs by the process of vertical gene transfer, so passing changes on from generation to generation, and they can occur by mutation, or if something has been acquired by the process of horizontal gene transfer, then it will be passed on to the next generation. Gene expression, which is transcription and translation. So the process of the DNA of a gene being transcribed into the RNA of messenger RNA and then being translated into a protein, a string of amino acids that folds into a three-dimensional structure that functions. Is basically the workhorse. The proteins are the workhorses of the cell. Now, regulation of gene expression can occur at the level of transcription, where the gene is turned on or off. Translation, so after the messenger RNA is made into protein, there can be modifications made to the proteins, adenyl groups added or phosphate groups added in order to modify its function, as well as protein function. We learned about enzyme regulation can occur either by competition or indirectly by a non-competitive inhibitor. So these are the ways that the bacteria tries, a single bacterium would try to be as efficient as possible in its utilization of its resources. And then recombination, which is the horizontal gene transfer, is the genetic transfer of these mobile genetic elements such as transposons or plasmids or genomic islands, or the accidental transfer of a fragment of DNA from another organism via DNA-mediated transformation, so picking up DNA directly from the outside environment, or by a virus that accidentally picks up some bacterial DNA from a previously infected cell and transfer it to the next cell that it infects. So I just wanted to go quickly over how bacteria reproduce their or go through DNA replication. They require a site for the start of the replication, which would be a recognition site for the enzyme DNA polymerase. It's called the origin of replication. As I mentioned, it occurs in two directions. So each strand of that double strand of DNA that's part of the chromosome can serve as a template to make a new strand. So in the diagram, the two dark purple strands serve as a template. They break apart, so enzymes help to unwind the DNA such that they can be separated. And there's continuous replication on the sense strand, and then in the other direction there's a discontinuous replication where you get these fragments of DNA called Okazaki fragments that are then ligated together by a DNA ligase enzyme. But basically in the end of this process, so the leading strand, it's continuous, the lagging strand is where you get those fragments. Basically in the end you've got an exact copy. So you started with one chromosome and you end up with two. And then the rest of the process of binary fission will continue as far as synthesizing the new plasma membrane and cell wall that will result in two separate cells. So right before they separate, there's a termination site where all of the machinery that's necessary for this process will detach from the DNA, and then the two strands will separate physically. So in the end, you've started with the parent DNA and you end up with two chromosomes, each of which have one strand that's the parental, so the darker blue or darker purple strand, and one newly synthesized strand, which is the pale purple in the diagram. Any questions before I go on to how the genes within this genome are then transcribed and translated? Okay. So transcription requires a key enzyme of the RNA polymerase. So DNA polymerase is for DNA replication. RNA polymerase is for making messenger RNA. And that involves the introduction of a new set of bases. So for every base that's in the sequence of DNA, there's a complementary base that's introduced in the RNA. So how did the DNA and the RNA bases pair up? So what are the DNA bases that I'm talking about? Yeah, adenosine, thymine, cytosine, and guanine. Okay, so this is the DNA. How does it change when it's RNA? So if this was the sequence in the DNA, what would I see in a sequence of messenger RNA? Yep. Uracil, not thymine. Okay. What would I get here? Yeah, adenine. Good. Yep. Good. So this would be the messenger RNA. And then another type of RNA is two other types of RNA and are important in the next step of translation. So for translation, you need transfer RNA to transfer each amino acid that's required in the sequence of the protein. And you need what other kind of RNA? That's part of the key molecule where translation occurs, or the macromolecule. Ribosomal RNA, which is part of the ribosome. So it's part protein, part RNA. So on this molecule of the ribosome, the messenger RNA, the ribosome will sit down on that messenger RNA. Each triplet of bases will be a codon. And each tRNA has an anticodon that will recognize this codon. So for example, the anticodon here in the tRNA would be AUC. Okay? So, but the anticodon is associated, so there are many different types of tRNA with different anticodons. And each one also carries at this other end an amino acid. So for example, the anticodon shown here, GAC, the codon is GAC, the anticodon is CUG, and it's encoding can't read it. Aspartate. Aspartate is the amino acid that's being carried by this particular tRNA. So in that way, you'll get these codons will give you a sequence of amino acids that are linked together covalently by the ribosome during the process of translation. I'm just going to take a break here to show you some animations of these processes just to help you review.
By formation of an initiation complex consisting of the smaller ribosomal subunit, the first amino acid tRNA, and messenger RNA. The larger ribosomal subunit then joins the complex. Proteins called initiation factors are also involved, but are not shown. The 70S ribosome has two sites to which transfer RNA carrying amino acids can bind. One is called the peptidyl or P site, and the other is called the acceptor or A site. The initiating transfer RNA carrying its amino acid binds to the P site. A transfer RNA that recognizes the next codon and carries the second amino acid then moves into the A site. The amino acid carried by the transfer RNA in the P site is then joined to the amino acid carried by the transfer RNA that just entered the A site. The ribosome now advances a distance of one codon, and the first transfer RNA is released. A transfer RNA carrying the next amino acid now moves into the A site, and the ribosome shifts down by a distance of one codon. This is followed by the two amino acids on the transfer RNA in the P site being transferred to the third amino acid. The ribosome continues to move along the messenger RNA, and new amino acids are added to the growing polypeptide chain. Elongation of the polypeptide is terminated when the ribosome reaches a codon that does not code for an amino acid, called a stop codon. The ribosome dissociates into the smaller and larger subunits, and the messenger RNA and protein are released.
Okay, so remember that within the ribosome will recognize, or the tRNAs will recognize, three types of codons: a start codon, UAG, amino acid encoding codons, which are usually more than one per amino acid, and then a stop codon, one that doesn't encode for any particular amino acid. At which point the complex will fall off and you'll have your translated primary sequence of amino acids, which will fold into its secondary and tertiary structure of the functional protein. So in bacteria, as the animation was discussing, there's not the eukaryotic ADS ribosome. Remember, we talked about the ribosome structure is a little different in bacteria. They have a 70S ribosome, and the 70S is just a measure of how it sediments when you centrifuge it. So it's just a term used to distinguish the two. So we know they're different because they sediment differently when they're centrifuged. But it reflects a change in the structure of those two molecules. In addition to having a different ribosome, there are several differences that occur in prokaryotes such as bacteria during the process of transcription and translation, because there are no internal compartments or organelles within these cells. Also, the organization of the chromosome or the genome is a little bit different as well. So mRNA does not need to be processed because it doesn't need to be relocated out of the nucleus. So usually in eukaryotes, they get a cap and a tail. It doesn't occur in bacteria. Also, the genes are not interrupted, so there are no introns and exons. There's just the coding, just the genetic code in each gene. It doesn't need to be spliced in any way or altered in any way. So the mRNA will directly encode a protein without having any kind of modifications to it, which enables the process of translation, because we don't need any of these modifications that are required in eukaryotes. The process of translation can actually occur as soon as the messenger RNA is synthesized. So translation can occur even during the process of transcription, even though it hasn't completed, as long as those parts of the mRNA that are completed, the ribosome can find that start site, that start codon, can form the complex, can start adding the amino acids. Also, you can get, so here's in purple is the DNA, and then you have many RNA polymerases making a transcript, the mRNA, which is in this very pale blue. And then the red are the ribosomes that are working to make the proteins. And they didn't actually show the growing protein chain in this cartoon. But there would be a growing chain of amino acids coming off of each of these red images. Each of the ribosomes. So that can occur very rapidly, more rapidly than in a eukaryotic cell. Also, the mRNA may encode for more than one gene. As I mentioned in the organization of the operons, you get one mRNA, but in the end you get 3 proteins. In the case of the lac operon, you get 3 proteins. So that's what is called polycistronic. And then the amino acids are cleaved apart to form those three mature proteins in the end of the process of translation. This is actually an electron micrograph of what's depicted here in the cartoon. You can see these darker black circles are the ribosomes. This very pale strand, gray strand here is the DNA. So it's occurring at many sites, many different genes simultaneously.